You may start.
Okay, so a very good morning to Doctor and to my fellow friends. Today, our Group 7 and 8, will be presenting two topics from re-emerging disease, which is polio and Japanese encephalitis. Next. So, what is re-emerging infections? Re-emerging infections are disease that are used to be under control but are now coming back again because protection or prevention has weakened. Yeah. Okay, so for polio is re-emerging because it was almost wiped out by vaccines, but now new cases are appearing again in places with low vaccination, conflict, or poor access to healthcare.
Okay, so polio has existed for thousands of years. Evidence of polio-like paralysis appears in ancient Egyptian artwork from around 1400 BCE. In the early 1900s, scientists identified the polio virus, and major epidemics occurred in Europe and US. Improved sanitation surprisingly increased outbreaks because children were no longer exposed early enough to develop immunity. A major breakthrough came in the mid 20th century. Jonas Salk created the first inactivated vaccine in 1955, and Albert Sabin introduced the oral vaccine in the early 1960s. These vaccines gave the Global Polio Eradication Initiative was launched. At that time, polio paralyzed about 350,000 children each year. Mass vaccination has since eliminated polio from most countries. Today, while polio remains endemic, vaccines and global cooperation have nearly wiped out a disease that once caused widespread paralysis. Okay, that's all.
Next is global trends of polio. So poliomyelitis cases have been decreased by more than 99% worldwide since 1988. In 1988, the estimation 350,000 uh, cases annually in 125 plus countries. And today, polio remains endemic in only two countries, which is Pakistan and Afghanistan. Two poliovirus types, WPV2 and WPV3, have been eradicated globally. WPV1, white poliovirus type 1, is now the only wild type still circulating. So the reason polio persists in this area is because of the conflict zones and inaccessible regions, mobile or displaced population, immunization gaps, and difficulty maintaining surveillance in remote areas. Okay, next. Okay, so based on these maps, there are two countries with red color region, which indicates WPV1 endemic. Okay, next. In Malaysia itself, Malaysia was polio-free since 2000. And in Sabah, in 2019, CVDPV1 detected in an infant and environmental samples. The outbreak linked to low immunization coverage and cross-border movement. In 2019 to 2021, mass vaccination campaign is organized. All children below 13 years old get vaccinated, and over 2.5 million OPV doses deployed with WHO or UNICEF support. In September 2021, Malaysia declared the outbreak over. Currently, no active polio cases, but our country remains at risk and maintains strong surveillance. Okay, next.
Okay, now we are moving on to factor contributing to the spread of polio. First, we have fecal oral transmission. This is due through contaminated food or water. Next, we have inadequate sanitation and hygiene. This includes going to areas with poor sanitation, use of unsafe water, and suboptimal hygiene practices. Moving on, traveling and population movement. International travel from endemic countries can reintroduce the virus into areas where it was previously controlled, especially if the population immunity is low. Then we have low vaccination coverage. Reduced immunization rate will increase the susceptibility for outbreak to occur in the population. And lastly, environmental and socio-economic limitation in low-middle income countries. This is due to lack of infrastructure, resources, surveillance, and political commitment in low and middle income countries, which hinders control and increased transmission of the virus. That's all. Next.
Okay, now we look at the surveillance and control measures. Malaysia was polio-free for 27 years until December 2019, when a CVDPV1 case was detected in Sabah. The virus strain was genetically linked to the Philippines, which triggered an immediate response from the Ministry of Health. Malaysia strengthened AFP surveillance, searched actively for children with acute paralysis, and carried out environmental surveillance, including the sewage sample in Sabah and Labuan. Stool testing among healthy children was also conducted. Malaysia maintained the WHO benchmark of at least one non-polio AFP case per 100,000 children, and by August 2020, four AFP cases and 25 positive sewage samples were detected. For the vaccination campaign in Malaysia, to contain the outbreak, Malaysia launched supplementary immunization activities for all children under 13. The campaign used BOPV and MOPV2 supplied by WHO and UNICEF for more than 2.5 million doses were delivered through clinics. Next slide. Globally, WHO leads the Global Polio Eradication Initiative. The main goals are to eliminate wild poliovirus in countries like Afghanistan and Pakistan, and to stop all DPV outbreaks worldwide. For outbreak response, during outbreaks, WHO recommends rapid vaccination using MOPV2 or BOPV, depending on the virus strain. Countries are required to conduct a large-scale SIAS, and WHO now deploys MOPV2, a newer and more stable vaccine. For surveillance, country must maintain strong AFP surveillance to detect paralysis cases and environmental surveillance to track poliovirus in sewage. For lab networks under WHO confirm any detected virus. WHO goal: WHO aims to remove OPV from the routine use and shift globally to IPV-only schedules, which helps prevent vaccine-derived outbreaks. For the regional action, WPR or SCAR, in the Western Pacific and Southeast Asia regions, the strategy is to keep more than or equal to 95% vaccination coverage, continue strict AFP and serious surveillance, vaccine travelers from outbreak areas, and conduct SIAS wherever immunity gaps appear. For WHO support to Malaysia, WHO support Malaysia by sending experts to Sabah and Labuan, supplying MOPV2, strengthening the cold chain system, help run communication campaign with UNICEF, who also monitor vaccination coverage and surveillance quality from start to finish. That's all for surveillance, and move on to Japanese encephalitis.
Now we move on to Japanese encephalitis. Japanese encephalitis is an acute viral infection of the central nervous system, and it is caused by Japanese encephalitis virus. Japanese encephalitis virus is transmitted mainly by Culex mosquitoes, and Japanese encephalitis is re-emerging due to expanded mosquito habitats, climate change, agricultural changes, human movement, low vaccination coverage, and weak surveillance. This allowed the virus to spread into new cases.
Next, for the origin and history of Japanese encephalitis. Japanese encephalitis is first recognized during the late 19th century in Japan and is known as recurring summer encephalitis. There is a major epidemic during 1920 for Japan with more than 6,000 cases with high mortality. It is called Japanese encephalitis because it was first identified, studied, and documented in Japan. The Japanese encephalitis virus was first isolated by a Japanese scientist and is classified as mosquito-borne flavivirus. So next, it is transmitted by Culex trichiniorhynchus, and its amplifying host is pigs, and the reservoir are wading or wet waterbirds. Humans are the dead end host where the virus cannot be transmitted any further. And the Japanese encephalitis, it spreads across East and Southeast Asia, mainly because it is driven by rice cultivation, irrigation, and pig farming. It became epidemic in more than 24 countries, including Malaysia. So next, first inactivated vaccine produced and implemented is in 1950s Japan, causing the cases to drop drastically. In China and Southeast Asia, from 1970s to 2000s, large-scale vaccination were implemented using inactivated and live-attenuated SA14-14-2 vaccines. In Malaysia, Japanese encephalitis was documented since 1950s and now are endemic in Sabah, Sarawak, and pig farming regions. Therefore, vaccinations are implemented in those high-risk areas. So that's all for origin and history.
Moving on to the global trends of Japanese encephalitis. Japanese encephalitis causes 68,000 clinical cases annually worldwide. The case fatality rate is around 20 to 30%, where the survivors often have long-term neurological complications. Japanese encephalitis is endemic in 24 countries, including India, Nepal, and Bangladesh. Transmission peaks during monsoon and post-monsoon seasons, and children under 15 years old are most affected in endemic regions. So the reasons why Japanese encephalitis persists in these areas is because of extensive rice cultivation and pig farming. Besides, climate factors such as monsoon rains and warm temperatures favor the mosquito proliferation. Lastly, there is a low vaccination coverage in rural and remote areas. Next slide. So this is a photo showing the endemic countries in 2021 and 2022. Next slide.
Now, let's take a look at the factors contributing to the spread of Japanese encephalitis. For the first factor, which is geographical and population factor, it is concentrated in Southeast Asia, including Thailand, Indonesia, Myanmar, and Western Pacific, such as South Korea, China, and Japan, due to the long rainy or wet seasons, extensive rice cultivation, and high density of pigs and water. Rural and pre-urban settings also the perfect spread for Japanese encephalitis due to rice paddies is a perfect mosquito breeding sites in village area. Irrigation system leads to stagnant water. Pigs also often kept close to home, and water birds are common in wetlands. For the next factor of spreading, we're gonna focus on the mosquito population, which commonly caused by the Culex tritanurhynchus and other species like Culex vishnui due to their zoophilic feeding behavior, where the mosquito prefer to bite animals like pigs, cattle, and goats, which keeps the virus circulating between animals that act as server. The mosquito also active mainly at night, where it increase risk for night shift worker and people who sleep outdoors. Some mosquito species can pass the virus from adult female to their eggs. The next factor, which is pig farm development, pigs is a major amplifying host for Japanese encephalitis because more pig farms will increase higher risk of pig mosquito transmission, and it also increase the risk of pig farm worker. Japanese encephalitis can still infect pigs even if the population is small, and it also may spread through oronasal secretions in pig, especially in areas with short mosquito seasons. Lastly, we're going to take a look at the migratory and wild bird in Malaysia. Birds are reservoir hosts for Japanese encephalitis. Migratory birds follow East Asian Australasian flyway and land in Malaysia, such as kapar, parit buntar, sabah, and pulau bird. These birds feed in paddy fields, which is the same site where the vectors is breed, and it also support Japanese encephalitis circulation.
Moving on to the surveillance of Japanese encephalitis, the goal is to detect early, monitor trends, and to guide intervention that focuses on humans, vectors, and animal reservoirs. So the first one, we can do case surveillance, such as the use of acute encephalitis syndrome surveillance and mandatory reporting of suspected cases. We can also do clinical and lab confirmation by IgM ELISA, and Sentinel hospitals can monitor the Japanese encephalitis activity. Next slide. We can also do laboratory surveillance such as ELISA and PCR. And also we can do vector surveillance to monitor Culex population density, identify breeding sites, assess insecticide, and predict high-risk seasons. Next slide. Lastly, we can do animal surveillance to monitor the pig seroprevalence and to observe piglet infection clusters. Next slide. So there are a few control measures of Japanese encephalitis. The first one is through vaccination, where we target children in endemic rural areas, travelers to endemic regions, and residents that is near pig farms or rice paddies. There are two types of vaccine, which is live attenuated and inactivated. So the live attenuated is called Imoject, and the inactivated is called Isiaro. Okay, next slide. We can also do vector control, which is the use of insect repellents or sleeping under mosquito nets, installing window screens, and avoiding outdoor exposure during dusk and dawn. We can also do mosquito management to target the correct mosquito. The measures include larviciding and fogging, draining stagnant water, and managing rice field irrigation. Next slide. The last one we can do is community engagement and education, such as awareness campaigns that focuses on educating rural populations on Japanese encephalitis symptoms and transmission, the importance of vaccination and mosquito avoidance practices. And that's all for polio and Japanese encephalitis. Thank you.